So our next speaker on the agenda is Roger Lowe. Roger is one of the recipients of the Hope Foundation Impact Award several years ago, and we wanted to highlight some of his work as kind of an oral progress report. Roger trained under Joanne Massagay at Memorial Sloan Kettering, and Joanne is a Howard Hughes Medical Institute investigator. He did his residency at UCLA and has stayed on staff there working closely with several people, Owen Whitty as well as Tony Rebaz, who will be our last speaker today. He's been funded by the NCI, NIH, Sidney Kimmel Foundation, Stand Up to Cancer, AACR, and the SWOG Hope Foundation, most importantly, the SWOG Hope Foundation. This past year has been remarkable. He had 2 papers in Cell, and in the past few years has had papers in Nature, PNAS, Cancer Cell, Nature Medicine, Nature Genetics, New England Journal of Medicine, etc. So, Roger, we're happy that you are a recipient of the Impact Award because you represent us well, and we look forward to your talk.
Thank you, Lee, for that very nice introduction. So I definitely want to thank the Hope Foundation for funding a part of our work, and also want to thank you for the opportunity to share some of our progress over the recent years. I'm going to try to squeeze in 6-7 years of work in 10 minutes, so good luck with me. Okay, so over the recent past, the therapeutics in advanced melanoma has really entered the double and triple combinatorial era, and I believe the study of resistance has been driving the landscape here and will continue to drive the landscape here. Our work is initially focused on the mechanisms of resistance to BRAF and the MAP kinase pathway inhibitors, although as we dig deeper and deeper into this problem, we've started to stumble into some unexpected findings, one of which is the profound influence that targeted therapy against mutations may have on the tumor immune microenvironment. So I'll speak to that sort of briefly toward the end of my talk, and then Tony, toward the end of the session, will speak a lot about the other cornerstone of melanoma therapy, the PD-1 antibodies. So let me start by looking through at this problem through the lens of genomics first. This is a study that we published almost 6 years ago, really providing the first genetic evidence that MAP kinase reactivation, again, sorry, Lee, I'm showing this very simplistic diagram here, but MAP kinase reactivation really mediates the main type of mechanisms to acquire BRAF inhibitor resistance. And so this type of studies really justify the combination of BRAF and MEK inhibitors in overcoming or suppressing resistance. As we come to realize the problem in patients is much more complicated, there are at least several reasons why the BRAF and MEK combo is not sufficient. I'm just going to highlight a few things on this slide. First thing, there are a series of mutations that turn on the MAP kinase-driven program and that are actually downstream of BRAF and MEK, one of which I'm showing here is a CDKN2A loss-of-function mutation. You also have a slew of mutations in the PI3K AKT pathway, and these provides compensatory survival and growth signals to the cancer. And on top of that, these 2 core resistance pathways, mutations in these pathways occur within the same patient, frequently within the same tumors. And among these resistant tumors, what you see is a very divergent type of phylogenetic relationship. And all this is to say that the cancer has gained the upper hand as it acquires resistance, becoming a more fit cancer. And then the functional importance of the PI3-AKT pathway is further sort of corroborated by these studies where we show that in early during therapy, there's an adaptive upregulation of this pathway, converging on AKT through a cell autonomous mechanism shown here on the top, and on the bottom here, another study actually carried out by my PhD mentor, Joanne Massagay, showing a non-cell autonomous mechanism through the secretion of factors that are induced by the therapy itself. And as we look into the tumors that have escaped from BRAF plus MEK combinations, what we found wasn't too much of a surprise, but actually sort of revealed a lot of therapeutic opportunities in these studies. One of which is that the genetic reservoir of the cancer is certainly not exhausted by the combination of BRAF and MEK inhibitors. And what we find is that there are a lot of these unusual genetic variants. I'm going to highlight a couple in the MAP kinase pathway, but the idea is that these alterations augment the signal zones in both of these core resistance pathway. An example I'm showing here is a signal zone resulting from these unusual genetic variants, one of which is BRAF ultra amplification. This is copy numbers in the range of 50 to 80 copies per tumor. We have a double mutation in BRAF and MEK, which is virtually not encountered in the TCGA studies. And the result of these are certain properties of signaling to which we can identify specific vulnerabilities. For example, we have this signal zone consisting of BRAF, CRAF, and MEK, whereby the amplified or overexpressed BRAF interacts with CRAF through a back-to-back interaction. And in the terms of this genetic context, the BRAF actually interacts with MEK through what's called a face-to-face interaction at a specific protein-protein interface. Another interesting finding that we reported here is the direct result of these altered or augmented signal zones. What happens is that if you take both of the inhibitors away from these tumor cells, resistant tumor cells, you see a hyper or super activation of the pathway, resulting in paradoxical loss of fitness to these cancer cells. So I think the understanding of this process we term melanoma drug addiction is potentially a therapeutic opportunity. So if we can lay out all the molecular steps that leads to this and lay out all the variables that leads to variable degrees of this phenotype. So I mentioned at the outset that the molecular lens to which we look at this process doing genomic analysis is no longer sufficient. We can no longer ignore some of the major players inside the tumor, the gorilla, the 800-pound gorilla, as well as the elephant, so to speak. So this is the tumor's ability to transition between different states via epigenetic means, and the ability of these altered cell states to impact the tumor microenvironment. And I'll speak to that shortly in the next couple of slides. Again, this is a study where we try to attempt to look at beyond the genomics, since we are sort of puzzled by many of the cases clinically where genetic analysis were insufficient to explain the resistant phenotype. And so this study laid out many of the sort of non-genomic and immune alterations are highly recurrent, in fact, much more recurrent than any single genetic mutation in any single gene. And in fact, this epigenetic basis really contributes to a whole multitude of hallmarks of cancer alterations in these resistant tumors. I want to just highlight a couple of things down here. One is in the tumor cell compartment. What we see is that these non-genomic alterations leads to a reduction of MAP kinase pathway addiction, so to speak, sort of changing the cancer's addiction to MAP kinase pathway into other alternative pathways, and a reduction in the sensitivity of these cancer cells to apoptosis. And we mapped out specific alterations in signaling that leads to these changes in upstream and apoptotic thresholds. And in the tumor immune compartment, what we show is that the highly recurrent changes in the expression of the antigen presentation machinery likely related to the loss of CD8 T cells and potentially their function as well. So with the knowledge that there's this non-genomic process that results in an alternative form of resistance, one which we term MAPK redundant, one that's not sensitive to this vertical pathway suppression approach of the MAP kinase signaling molecules, we started to wonder whether we can dissect into the genesis of this process during the course of treatment. Of course, we look into the early tumors that are persisting in the patient. And this is a study where we went on to analyze cell line models as well as tissues derived from patients before therapy, as the tumor regresses in the patient, as the tumor persists in the patient, and undergone a sort of parallel transcriptomic methylomic analysis of these processes. And what we found is that, in fact, early on therapy, as the tumor shrinks, there is already extensive evidence that the tumor phenotype is changing, is switching. And one of the most important processes we identify here is a mesenchymal invasive transition. It has a very clear epigenetic basis to this process, which is also highly linked to a series of other properties of the tumor, including angiogenesis, ECM remodeling, wound healing, and even immune-resistant genes being dysregulated. So this is really the first evidence that tumor in regression undergoes dynamic transcriptomic metabolomic changes, which is starting to blur the line between what a cancer cell is and what is a stromal cell in the tumor microenvironment. And one of the importance of the set of processes I just identified here, which is sort of induced in mass, in bulk, by a single functional perturbation, the addition of a MAP kinase pathway inhibitor. We also try a variety of different insults to induce stress in the cancer cells, and they can indeed induce these set of processes as well. But one of the most clinically evident importance to these processes is underlying here when we analyze the transcriptomic signature that may be related to innate anti-PD-1 resistance in melanoma. So let me just go into that briefly in the next slide. This is an analysis of gene signatures, differential enrichment of signatures across the two groups of melanoma tumors, one non-responding and one responding to PD-1 therapy. We call these signatures are red here, enriched on the left in the non-responding tumors, the IPRES signature for innate anti-PD-1 resistance. And there are two main sort of take-home messages in this slide. One is that, as you can see, this signature is overrepresented on the left and underrepresented on the right. And the other thing is that when this is overrepresented, it appears to be co-enriched across the same set of tumors. And this just sort of harkens back to the earlier study where we show that with a single functional perturbation, addition of MAP kinase pathway inhibitor, you can actually induce a whole series of these processes, suggesting a common transcriptomic basis of this. And again, you can see these MAP kinase-induced signatures are present, underlying discrete processes in this whole set of signatures, along with other independently derived signatures that we analyzed. And that this pattern of enrichment on the left in the innately resistant tumors is not happening by chance, and this is sort of supported by this analysis across 4 different metastatic melanoma RNA-seq cohorts, showing again that co-enrichment of the signature, this IPRES signature, is very stable, consisting of about 30% of each cohort in metastatic melanoma, being slightly lower in primary melanoma. And then intriguingly, as you look across other cancer histologies, you can also see this very stable transcriptomic subset present in different proportions, but in pancreatic cancer, as high as 80 to 90% of the tumors shows relative enrichment of these signatures. So with that, I just want to recap, and my time is running out, with a couple sort of main points I've covered. So recurrent genetic drivers of acquired or late resistance, it largely hyperactivates 2 core resistance pathways. Transcriptomic, metabolomic, and immune alterations are highly recurrent in late resistance. And as we look earlier during therapy, these processes probably result from a set of processes that are induced by the MAP kinase pathway inhibitors, causing tumor cell intrinsic changes that has potential to modulate the immune microenvironment. So with that, I want to thank all the people that contributed to these works, including the postdocs and the graduate students in my lab, my collaborators, Tony Ribas especially. He's been my steadfast collaborator for almost 10 years now, starting when I was a clueless postdoc in Owen Whitty's lab. And then Bartosz Chmielewski, Alan Algazi from UCSF, Jeff Salzman, Doug Johnson have been some of my best collaborators from Vanderbilt. And of course, the Hope Foundation funding that came in very handy. Thank you.